Alright, this video is for Biology 234, and we're going to be discussing simple and gram stain. So first we'll discuss differential staining. Differential staining procedures such as the gram stain and endospore stain use two or more reagents to detect dissimilar aspects of cell physiology. Using these techniques provides important information regarding differences in cell structure. The differential staining techniques used in the lab begin with a bacterial smear. So now we'll discuss the gram stain. The gram stain was invented by Hans Christian Gram in 1884. The original purpose of the staining technique was to stain bacteria in lung tissue samples. We now use the gram stain as a way to differentiate between cells that have a thick peptidoglycan containing cell wall, which would be gram positive, and those that have a LPS containing outer membrane, which are gram negative. Fresh cultures should always be used for the gram stain, preferably those in log phase, but early exponential phase may work as well. Cultures that are too old do not stain well and may not stain as expected. 4 reagents are used in the gram stain procedure. The first reagent is crystal violet. So I got the gram stain kit out. This is crystal violet. Crystal violet, called the primary stain, stains the peptidoglycan cell wall of gram-positive bacteria and stains the outer membrane of gram-negative bacteria. After the first step, all bacteria will appear purple. The second reagent is Gram's iodine. This one's empty. I'll have to grab another one. Gram's iodine acts as a mordant, which is a substance that allows the primary stain to be retained without the peptidoglycan cell wall of gram positives. Specifically, the crystal violet and the iodine form a molecular complex that is more easily trapped within the peptidoglycan. In addition, iodine kills bacteria cells and denatures cellular proteins. So after this step, both gram-negative and gram-positives will be purple. The third reagent, called the Decolorizer, is 95% ethyl alcohol. Acetone may also be present. This removes the crystal violet from the gram-negative cells, in part by washing away the outer membrane. This is the most critical step of the procedure. When added to the gram-positive cells, the alcohol dehydrates the peptidoglycan, shrinking it, trapping the crystal violet iodine complex. It is important to pay particular attention during the decolorization step. It's possible to over-decolorize or under-decolorize your smear. Gram-positive cells will appear as gram-negatives, and gram-negatives will look like gram-positives if this occurs. When done correctly, gram-negative cells will appear colorless, and gram-positive cells will appear purple after this step. The fourth reagent is safranin, which looks like this. Which is called the counterstain. Counterstains produce a color that is easily differentiated from that of the primary stain, and also target an aspect of cell physiology that differs from the target of the primary stain. In this procedure, the safranin stains cell membrane of gram-negative cells, turning the colorless gram-negative cells pink, and gram-positive cells will remain purple after this step. During any staining procedure, a reagent is washed off before proceeding to the next reagent. In the gram stain, slides are rinsed with cold running water between reagents. After one reagent is washed off, the next reagent is added immediately. The slides should not be allowed to air dry in between stains. Alright, so now we're ready to begin the gram stain procedure. You will use the gram stain to determine cell shape and whether your organism is gram positive or gram negative. To use time effectively, you should stain all 3 smears simultaneously. So I'll put all of my slides onto the slide rack. Step 1, read this procedure thoroughly and then gather all required materials. So these are all of the gram staining reagents. And then I have a squeeze bottle of DI water to rinse between reagents. 2, put on gloves and protective goggles. 3, place your smears on the staining rack over a sink. Do not leave clothespins on your slides. 4, carefully add crystal violet to cover the smear and let it stand for 1 minute. Then you just want to cover any spots that you missed. I know it seems excessive, but you don't want to have mixed results. So now we'll wait 1 minute. Alright, it's been a minute, so now we'll move on to the next step. It says, gently rinse the excess crystal violet from the slide with cold running water. So just rinse it. Alright, now we're at step 6. Carefully add grams iodine to cover the smear and let that stand for 1 minute. Alright, so we'll let that stand for a minute. Alright, it's been a minute, so now we'll move on to the next step. We're at step 8. Pick up your slide and hold it at an angle over the sink. And then step 9 is carefully rinse the smear with decolorizing agent. Allow the reagent to run over the smear and drip off the end of the slide. Stop decolorizing when the runoff just starts to run clear. It'll be dark blue when you start. Alright, so we'll start with this one. Alright, it's starting to run clear-ish. And we'll move on to this one. So I just wanted to point out that this one, after the decolorizer, isn't purple anymore. But this one retains some of the purple. I'm not sure if you can see that. That will probably help. We're at step 11. Carefully add saffron to cover the smear and let stand for 45 seconds. Right, so now we'll wait 45 seconds. Alright, so now we are at step 12. Gently rinse the excess saffron from the slide with cold running water. And then step 13, rewrite labels with a wax pencil if you inadvertently rinse them off with the reagents. And then it says, place the stain slides between sheets of dry bibulous paper to remove excess water. Do not press the pad down with your hands. You may crack your slide. So we'll grab some bibulous paper, which looks like this. And we'll just place our slides on it. Dry. And then we'll put another one on top. We're at step 15. When the majority of the water is removed, do your smears under the oil immersion lens, and then record your observations and conclusions in the results section. And then when we're done, dispose of the slides in the bleach water. All right, so now we'll move on to looking at them under the microscope. This is the E. coli, I believe. Yeah, it's the E. coli under 100x. So record any observations that you have. And we can move the slide around maybe. But it was pretty uniform throughout the entire slide. And now we'll look at the staph epidermidis under 100x. All right, so this is the staph epidermidis under 100x oil microscope lens. And I think it's, yeah, that's about as focused as I can get it. Yeah, the part over to the left, you can make out the shape and arrangement of the bacteria a lot better. So now we'll look at unknown 9. So this is unknown 9 under the 100x. And this one also looks like there's multiple layers. Well, this looks like a better part of the slide.